So last speaker, Jeff Milton. He is the founder and CEO at Lafora Labs, a company focusing on RNA therapeutics technology. Jeff is the former head of data sciences at Arcturus Therapeutics, contributing to rare disease programs. He was also the director of functional genomics and drug discovery at Ionis Pharmaceuticals. Jeff.
Thank you for sticking around. So when I was at Ionis, we used to have this thing called a program review, and it was an annual, it was an annual thing that we used to have to present to the entire company. It was a much bigger room. It was full of people. Stan Crook would sit right in front of you, and all the senior leadership would sit right in front of you, and your boss would sit pretty close too. Sort of feels like that right now because there's my old boss right there, Chris Hart. So. And Tamar was also in our group as well. She's my boss now, but I'm going to take us out a little bit. I have a little bit of a different take on the space of basically a combination of money and how do we create a competitive landscape around rare disease and the interaction with patients. And I'm going to use Lafora disease as a case study for this because I think it's the canary in the coal mine, to use Chris's parlance, for something that's coming that we need to be aware of. And so Lafora Labs. And so what I'm going to start off with is actually a story. When I was young, I lived in Berlin, and back in those days, it was a divided city, and it had a wall around it. And it took about 2 hours to drive from, so actually the West Germany there was British sector, French sector, and American sector. The big part, that's West Germany. It took 2 hours to drive through the Soviet sector to get to Berlin. So it was a walled city of freedom inside the Soviet sector. And inside Berlin was actually divided into several sectors as well. So there's the French and the British and the American sector. And I lived down on the bottom of the US sector. My father was in military intelligence. So we had a house actually in Potsdam, so in East Berlin, and or in East Germany, sorry, Potsdam in East Germany. So we used to cross this bridge called the Glenneke Brücke. And this is that bridge. And the one thing that was very compelling to me when I, as a young child crossing this bridge, and you can see it in this picture, there's a nice green section, and right at the exact middle, it turns black. And that's the border between West Berlin and East Germany. And the difference was that obviously there was care and there was civilization that progressed, and there was a civilization that didn't progress. And the way, as I grew older and understood the economy and how government worked a little bit, I understood the concept of private ownership versus common ownership. And in 1985, this is the Glenneke Bridge. The Soviet Union is on the other side. Now, the reason it was black, first of all, was because back in those days in 85, they used to heat their homes with coal. So you'd have a coal furnace in your basement. And then they would dump the ashes out in the dumpsters, and then they put those out in the street. So everything was black. And everything was just dark and covered. But in any case, what I'm trying to get to here is that in 1985, in the West Berlin side, you could have a BMW M6, 256 horsepower. And then in East Germany, it was a Trabant, 25 horsepower, and you were lucky to get it. It would take years to apply for this, and you only had a couple of colors you could choose from. So in rare disease, we need precision, we need high technology, we need an economy that breeds this type of technology. And so really in rare disease, we need something like the 1985 308 GT. And okay, some people might want to take Tom Selleck with it, but in any case, we need something that's very high with a lot of high precision. And so it's great to be in an environment that actually promotes profitability, competition. These are things that breed innovation. So we have to keep that in mind. And so I come from working many years at this company. Chris and Tamar were there with me. And this is a product of innovation and a competitive environment, is the platform that they developed. They started off with $5.2 million in VC funding. There were 5 other companies that started off in the same field, and they just duked it out. And that's the way it should be, right? So the one who wins is the one who moves forward. And now they're at a point where 16 RNA targeting drugs approved, the first blockbuster drug, Spinraza. They're expecting another approval this year from iPSC-3 GalNAc conjugate. All routes are enabled, broad distribution after systemic administration, targeted delivery to liver, beta cells, and pancreas. And what's really amazing is that now with this technology, over 250,000 people have been dosed, and 20,000, mostly kids, have been dosed intrathecally. So, and obviously there's a lot more that's going on. So what is this technology? And I have this up and sort of showing 2 different pathways here. The one on the left is the Ionis sort of what they focused on, what they developed is essentially this RNA-mediated knockdown. It's a single-stranded ASO, binds lots of create base pairing to a target RNA. RNA being there, essentially located at the point of transcription, cleaves this RNA, and then you have degradation of the RNA, so you don't create that protein. So that's the mechanism of action of this drug. So it's exactly what Chris was saying before. It's a matter of programming the sequences and understanding that. Well, siRNA is the same, somewhat the same approach. It uses different enzymes. It's a duplex of an antisense strand and a passenger strand, and it uses a RISC-mediated knockdown approach. And that's been the 2 main companies that kind of paved the way for that, as Ionis is the ASO, or the single-strand ASO, and then the alma is the one you think of with the siRNA approach. So now I'm going to jump into a disease that is fundamental to La Jolla Labs, because we actually started our company because of this disease. And it's a disease called Lafora. And there's a drug that was created for it. And it's a progressive neurodegeneration. This is Chelsea Gerber. There's a foundation called Chelsea's Hope. And this is a disease that once you are diagnosed with this, it is a steady decline into a vegetative state and then death. So first, to understand the disease, you have to understand a little bit about how glycogen, the metabolism of glycogen. So glucose enters the cell, glycogen synthase polymerizes or catalyzes the polymerization of this into long carbohydrate chains, single strand chains, and then a branching enzyme takes that glycosidic bond and moves it down and compacts it a little bit and keeps it into a very tight but soluble carbohydrate bundle. And then on the degradation or the breakdown side, glycogen phosphorylase pulls the, or debranching enzyme along with laforin pulls this off, and then it goes back into glucose and it's used for energy. So during the fed state, you create this glycogen polymer and then you consume it as you need it. It's a cache. Over on the right-hand side, you'll see that there's a diagram that shows glycogen in the context of, really, it's been linked to a lot of very fundamental biology. And that's kind of the roadmap that I like to look at coming from the point of view of rare disease, because that means that there's actually a lot of other avenues that you can pursue as far as a therapeutic. So when we talk about this long tail, I like to think that there's a long tail, but there are hidden spikes in that tail. And that's where you can have these rare diseases that have potential broad applications. And that's some of the stuff that we're doing at La Jolla Labs. So in any case, going down the path here to the therapy, this is just a look at wild type periodic acid shift. So it stains the actual glycogen deposits, Laforan knockout. The mutation can actually happen in two different genes, Laforan and Mellon. And as a result, you get these Lafora bodies deposited in the cytoplasm. And that's the pink or the magenta speckles. So you have the Laforan knockout and the Mellon knockout, and you can see that there. So the idea is that's the pathogenic part, is the accumulation of these Lafora bodies. So if you can essentially reduce the synthesis of this in a way that prevents the accumulation of these glycogen Lafora bodies, then you could potentially keep it in solution and prevent these Lafora bodies from forming. And it turns out you only need to knock down the synthetic enzyme or the enzyme responsible for synthesis about 50%. So that's the rate limiting step in the process. So I'll skip to this, and this is just kind of a schematic that highlights how this drug works. So there's a healthy state, and that is that you're producing the glycogen at the same rate that you're consuming it. The Lafora disease state is that it's obviously accumulating and you have aberrant Lafora bodies. And then the therapy and the therapeutic approach is to use a gapmer ASO that you can knock down the RNA of glycogen synthase 1 in a way that slows down the synthetic process of the polymer in a way that, and prevent the Lafora bodies from accumulating. And this is actually a drug that's so this sort of highlights how that mechanism works. So obviously looking at the central dogma, you have the DNA creates RNA, RNA creates protein, the protein goes off and does a function. In the middle there, you'll see the actual diagram. The Pac-Man is essentially the RNA-CH enzyme. It cleaves in the gap of this gapmer ASO, and then the enzyme or the RNA is degraded, protein isn't created. It turns out you only need 50% knockdown to make this work. And so I'll skip through some of this, but basically these are some of the experiments that were done to show that this actually does work in mice. And you can see that with, you can see kind of, actually this image isn't that great as far as the coloring, but you can see that the deposits are much less in the GICE1 ASO. So like I said, it's a progressive neurodegeneration. And this is, I think she was diagnosed in 2020, and this is her last year. And it's absolutely devastating disease. This drug was created by Ionis, and it really happens at the screening process. So they do the design and they screen, and then they patent it. And it's actually created by Tamara Grossman, who's sitting right over there. So I'm trying to speak for all the work that she did. But so she, so it's gotten to, it went all the way through to preclinical IND or pre-IND phase. And they got to a point where it wasn't financially viable as far as how they were going to move the asset forward. And this is a reality across the entire space. We all have to face this. And again, this is a company that's for profit, and they've got to make a tough decision. And, I think we were there when they made that decision. And so they tabled it. So normally in a pharmaceutical company, you have executive oversight over a particular program, and you have executive decisions and thought and strategy on where that program is going. Once you table that structure, once you table that asset, it goes into a separate bin. And that bin is a bin that's basically business development. It's transactional. It's, okay, we're going to liquidate, we're going to find a way to move this forward. And that's, at that point, you don't have a strategy other than liquidation and moving it forward. And you don't have executive thought or leadership over that necessarily. And that's all pharmaceutical companies right now, especially big pharmaceutical companies. That's not isolated in any way. So what you have is you have a drug that's gone all the way through to preclinical setting. It's at IND, it's ready to be dosed in the clinic. It's a very expensive clinical trial. They have to make a tough decision. And actually, to tell you the truth, we kind of agreed with that decision as far as a financial model. So the reason that La Jolla Labs was created was really to find that way to go past that point. But when you look at the landscape of where Lafora is right now, this is what you look at. And so this is a very difficult place for a small rare disease. So I should say the rare disease is about 200 to 300 patients. So it's in that long tail, and it's a very difficult space for any company. So what you'll notice is actually there are two companies that I've shaded them in blue. These are companies that actually have engaged with the patients, either through natural history or they participated in compassionate use programs. And what that does essentially for a small rare disease population is it essentially sucks the financial motivation out of the investors. In a sense, it's basically what happens is when you do rare disease therapeutics, the funding, the due diligence is a very high priority. They scrutinize that population. And the first thing they look for is who's already there. So when you start a rare disease program, if the NIH decides they're going to come out with a clinical trial and you've got a preclinical asset, you're kind of in trouble because it's all about getting to the patients. And if you have a company that has, or a couple companies in this case, that have essentially completely encapsulated those patients, you can have a good compound, but you're not going to get anywhere. So, and this was actually reflected by, it was actually a whole talk I went to yesterday. I was at UCSF Neuro Department, and Dr. James Levine presented the 6 guiding principles that drive innovation in rare disease. And that was one of his principal topics was the patients, getting to the patients in these ultra rare disease programs are a rate limiting step to any of these development programs. So like I said, La Jolla Labs was founded really to try to find a way to do discovery and to bring and deliver these things to the patients. And so what we decided was, there's this concept of essentially a boutique therapeutic. And that is, we do discovery, that's what we do. We create, people come to us, they say, I have a target, and we generate a compound for them, a lead compound for them. And we don't necessarily take it too far down the road, but what we found out is actually that people want to come, they have targets, they want to do a collaboration, they'll fund it. And a lot of times, as we've seen in this room, there's a personal connections. And that's changing the whole dynamic of rare disease. Because traditionally there is the big pharma, they sit in the buildings in their cubicles, they don't interact with the patients that much. There isn't a lot of energy, but I'll tell you, I speak to patients almost every day. I speak to patients and families every day. And there's so much more energy in them. And there's so much more information out there. And they're high functioning. They want to figure this stuff out. They have responsibility and they have needs instead of wants. And that's a very different driver. And that's why I think that from the point of view of a boutique therapeutic, you can have this personal connection that's really driving this whole thing. So what is it? It's basically a patient-driven, it's for-profit. I think that's by design. It has to be for-profit, modality agnostic. There has to be a succession plan, and I'll get to that in a second. There's low overhead, meaning it's a very targeted team. And there are built-in de-risking strategies. And I'll talk about that later. And this is very attractive to a whole investment group out there called impact investors. And impact investors are nonprofits the funds that they get go back into the nonprofit. So they invest with the idea of making money, but those funds go back into the nonprofit. And so, what we're trying to do is basically promote that ecosystem where we can spin off, we can have these companies that come to us, say, we want to develop a therapeutic. It's for, there may be an emotional connection to it, may not, but we find strategies for de-risking those assets in a way that's, okay, here's your target, and then here's how we can de-risk that by saying there are other targets that have broader indications. So we do this, I'll just highlight this very quickly, do a little bit of a deep dive, a little into the machine learning side of things, but we have a splicing model that helps us to do some of the design. We're focused on splicing. So this is an example of where Spinraza sits in our splicing models. We have attributes associated with the exclusion of exons and then the inclusion of exons. And it turns out Spinraza sits in this nice little pocket. It's almost like rational drug design. It just sits right in there. It's very nice. But taking that strategy or taking something like a platform that helps us do discovery and package that up into a team. And I like to think of this, if you've seen my N1C talk, I talk about it. I look at everything from the point of view of a competition where there's an offense, there's a defense, and there's a coach. The offense is, how do I increase demand? The defense is, how do I decrease costs? And the coach is just there to raise money. And so taking something like glycogen synthase 1, there are lots of other indications we can go after. So Lafora is our target audience or target patient population. But it turns out, and as Tamar published here in this paper, it's actually can be a drug that can be used for Pompe. But as you saw that map before, it can go well beyond that. It can go into, it's just a matter of good, smart, scientific minds sitting down and saying, what other areas can we go? What's the phenotype that we're looking for? Can that be, are there other mechanisms that we can go after that can de-risk this asset? And so getting back to our AI models for this particular target, we decided to kind of use this to generate our own glycogen synthase 1 inhibitor, but we're doing a different mechanism. We're going to actually slice out an exon and induce downstream stop codons. And this causes nonsense-mediated decay, and we get 50% knockdown with inducing nonsense-mediated decay using a splicing ASO. And it actually tiles, and we tile these over, it almost represents like a dose response to as you tile into the attributes that are inclusionary, it's cutting that out. We still have to validate this, so this is something we're working on. But in any case, that's the type of asset that we can kind of build into our system. And of course, my slides were a little out of order. I don't know what happened there, but the defense side of this is also to keep in mind that the FDA is a responsive institution. They're not going to proactively do things to enable rare disease. You have to approach them. And they have this as their formula: analysis of the condition, current treatment options, potential benefit, and risk. And when you're talking about something like Lafora, these kids are going to die. We know that. There's no real treatment that's out there for them. The potential benefit is they could survive. And what's the risk? It's minimal when you're considering the devastation of that disease. And of course, the coach is there to explore different programs. So the idea here is that instead of putting a drug into a business development bin, you keep it going with an executive team that can make executive decisions, and you move it forward. When it comes to communicating with the patient population, we actually have a program we're spinning out right now. It's targeted, it's allele-specific knockdown. It was recognized by very expert teams that came out of left field. They recognized this particular mutation as having broad indications. So they licensed it. And I had a problem, and that is that it was built for a very small community, and they licensed that. So the succession plan is built into this, and that we put a trust in place such that if that program is deprioritized or fails, it goes into that trust to ensure that that original patient population is protected, because I've already connected with them. I've already been working with them. So this is something that's new. We're kind of figuring our way through this whole thing. And so it's really important. I think that this is something that we're going to all kind of bump up against as we do genetic interventions on very rare populations. Thanks, Winston, for that term. I think that's perfect. The last thing I want to mention is, and this is basically getting to the end, but there are a lot of things that are changing in regulatory space. And there's also, I'm not sure how this will impact us, but it's something we all need to pay attention to, and that's the Promising Pathways Act. This is a law that was a bill that was proposed to this, introduced to the Senate recently by Senator Mike Braun from Nebraska, I think. In any case, I talked to his office. I got kind of the scoop on what the idea is, but basically it's a provisional approval for a rare disease drug, which basically before you even at the time of dosing, it's really kind of pretty far out there, but it's got a lot of support, and it's something you should read and understand if you're in this community and you understand the impact of what this is going to do. I can already tell you that NORD is not a fan of it, but I've looked at the list of who are supporting this, and it's a lot of nonprofits. So it's something you should definitely look at.
Why would ignore it? That is so.
I don't know. That's why I'm hesitant. I don't know. I would love to have somebody read it. I just read an article that they were not a fan.
How about global genes?
I don't know. It's Charlene, right?
Yeah.
I saw her yesterday. No, I didn't talk to her about it. Anyway, what we are is we are a discovery platform. We're trying to promote these things. And the one thing I'll end with is, companies like IXLs and Jolla Labs, we're opening the doors of big pharmaceutical companies. I mean, basically we're trying to promote these small targeted therapeutic avenues. And to the extent that we can support you through preclinical development, that's what we do for very targeted small diseases, small indications. And I have the names in the notes, but anyway, I just wanted to end with, I forgot, let's see. Anyway, recently diagnosed, this is her this year. And then Kyle, I believe his name's Kyle, was diagnosed in 2020, and he passed away last October. And, this is the difference. And the key here to remember is that when you stop a drug from being engaged in the strategy, the disease doesn't stop, it's progressing. So we have to always keep that in mind. And so that's, I think, our responsibility to keep that going. I'll end there. Thank you so much. Yeah.